Thank you very much, Jeremy. Just to check everyone can hear me. Was there a
Yes, yeah, we can hear you.
delay? So I'm delighted to be in South Africa. I'm here for a variety of reasons, which I'll tell about in a second. But I'm at the Liverpool School of Tropical Medicine, and I'd like to present to you some of the studies that we've been doing in Malawi on schistosomiasis, but also a bit of a broader picture. Some of it's going to be in the public health sphere, some of it might be in the One Health, but also I'll be touching upon some clinical aspects which might pique your interest for some of the manifestations you see with schistosomiasis, which is genital tract disease. Image on the left, these are cattle in Lake Malawi, obviously having water contact. So I'm going to try and tell you a bit about zoonotic schistosomes, which is the new aspect in schistosomiasis at the moment. Knowing it's a problem in Africa, but it's also becoming a problem in Europe. So schistosomes are actually being exported out through either return travelers or people visiting endemic areas, and it's causing a bit of a conundrum in Europe. And there's actually natural transmission of a type of schistosome I'll talk about in a second. So for those of you who know UK, there's two schools of tropical medicine. There's the London one, which my colleague here, John, did his master's at, but also there's a Liverpool School of Tropical Medicine. Liverpool's a bit smaller, it's older. I think it started just before the 1900s. And to tell you about, this is the old building, which you can see here. Originally, it was only half a building and it was just that site. And then from 1914 onwards, it got expanded so it balanced itself up. And then over the years, the footprint of the Liverpool School has actually increased massively. I took over in that glass building, and there's three or four other buildings which unify to form the school. It's also got its own university status now. So we do postgraduate teaching. We started to award our own degrees. This started about 2 or 3 years ago when they got awarding powers. There's 4 departments, scientific departments. I'm in tropical disease biology. There's a big snake venom unit in the school, which does a lot of anti-venom production as well as research on how to neutralize various snail toxins. And also there's a vector biology department, do a lot of work on mosquitoes, international public health, we do work on social science and international outreach, clinical sciences, do work in the hospital across the road, which is the Royal Liverpool. Also an education faculty, and we've just started running a new master's, which we'll draw attention to. It's called One Health and Tropical Disease. This is a photograph of some interesting work that we've done on Ngamba Island in Uganda, and there's a chimpanzee sanctuary. And because it's a small island in Lake Victoria, the chimpanzees have actually caught intestinal schistosomiasis. So what we're doing here is the chimp anesthetized. We're looking at the liver through ultrasound, looking at fibrosis. Oops. This was Alex, who's the vet who works with me. She's the director of the One Health degree. There's myself working with the vets, and we're taking biopsies from the chimpanzees. Is it because the battery might go down?
Sorry to interrupt, Jeremy. I hear that there's an issue with the sound. We're just going to try a headphone to see if that improves. Sorry, everyone.
No problem.
Because thanks. Is that an echo, or is it just the microphone's not picking up well?
I think.
Does the sound work a bit better now? Can you hear me?
Much better. Thank
you.
Oh,
much better then. Thank you, Charlotte. That's a save the day technology. Again, this is John, Charlotte. So it's nice to be visiting them. Yeah, where were I? The little photograph there is just showing that there's quite a bit of a connection. So chimpanzees don't normally get schistosomiasis, but in this managed setting they do. And it's just to point out, we've done a 14-year follow-up on some of these animals, seeing how the fibrosis has changed. Just to draw attention to the fact that, schistosomiasis, like several other zoonotic parasites, is really like the backbone of a lot of like One Health disease. So this September, we just started a new master's MSc, which I'm co-course director with Alex. And then just last week, the students at the Liverpool School get taken on a field trip, which is in Shropshire, and they get exposed to the type of parasites which are the distant cousins of those in the tropics. So they're doing dragging for ticks, looking at some of the, well, do small rodent trapping. We often spike some of the samples with tropical stuff to look under the microscope. I really enjoy that course, and you could have said I had the short straw by missing it, but I had a very good reason for coming to South Africa. The first reason was to attend the International Congress of Parasites of Wildlife. That was in the Kruger. That's with the South African Association of Parasitologists. And also it's to help supervise Bhavani here. And after the conference, we did a snail survey in Mpumalanga district. This is some of the team, and we're kind of discussing what type of snails we're finding, but also we're picking up cattle feces as we're discussing here with John. And I just want to point out, when we look at those feces, we might see eggs. This happens to be a fasciola or a liver fluke egg, and traditionally you would imagine this to be could only be Fasciola gigantica. However, you do get hepatica in South Africa. And the reason why this is I wanted to show you is because locally, there are two different types of snails now present in that area. The top one, which is Limnaea natalensis, is the usual host of gigantica, and it doesn't transmit hepatica, whereas there's an alien species, Pseudosuccinea columella, which has been kind of expanding its range for the past 20 years in South Africa. But that one is unusual because it can transmit hepatica and gigantica. So the only way to be sure if that egg that we saw in the feces was really a hepatica or a gigantica egg is to use molecular markers to identify it. So it's just to draw your attention to the knowledge of the snails, the knowledge of the water, knowledge of the environment, but also, traditional microscopy can get you to some type of algorithms of what disease you might find. But the application of molecular markers is really important, and it might cause some discussion of, how much human fasciolitis do you see in certain parts of South Africa? Anyway, bit more about One Health. It's not just medicine in animals. It's got some other components. And there was a special issue of the Lancet not so long ago, which was trying to get a kind of holistic approach. And there's a bit of a depressing statement here, which they used as a slogan. We have mortgaged the health of future generations to realize economic development gains, past and present. And in that special issue, there are these gradings of the planet in terms of how you quantify risk, not only to the environment, to the air, to the water. And I just want to point out one of the major ones is the fresh water. So it's in Sustainable Development Goal 14, life underwater. But I just want to point out those fresh water bodies, lakes, small streams, large streams, and so forth. They are pretty precious for a variety of reasons. So for the Schistosomiasis or the liver fluke or the lumen fluke domains, we're interested in those water bodies, the biology of the snails, the biology of the parasites, the way in which animals and people interact with that. So that's how we kind of frame One Health these days. But in reality, it's just rebranding, I suppose, the holistic epidemiology that we used to do for these type of parasites, ever since they were described by Lipa in the London School in 1916, which brought together, water-based control, water and sanitation, understanding the life cycle. It's a good example of a One Health disease, Schistosomiasis. So we should, and maybe on the fieldwork, we saw quite a few polluted sites. This is, some of the diseases we have, we cause it ourselves basically through pollution. And just to show that, plastic contamination, all sorts of stuff go into fresh water. It does cause issues with the water. One of the things that I've been doing, which is a bit of a surprise, of collaborating with colleagues at the school who are experts in AMR. And we had a PhD student, Angus O'Farrell, who was working with us. We sampled various freshwater snails because we were looking for schistosomes or liver flukes, but also we looked at the snail feces that come out. And if you genotype those feces and you search for AMR markers, you'd be surprised to what you find. So in United Kingdom, Uganda, Malawi, and Zanzibar, there's a tremendous amount of AMR markers and different types of bacteria, which can act as, I suppose, the snails can act as accumulators. So it's another way in which we think how water is tied to human health. AMR transmission in snails, for example, is another dimension that we're starting to explore. Now, I suppose this is a bit of a complicated diagram, but it might help understand where One Health fits into the broader scheme of epidemiology. In the center, we have like traditional control of diseases, which many of us are familiar with. So we have vector control, control of mosquitoes or tsetse flies, or even snail control. We have low-cost diagnostics, which help us understand, at scale, the disease burden. Some of the diseases we can control with access to drug, preventive chemotherapy, so mass drug administration. Also, depending on what the repertoire of the drug is, we can sometimes get parasites that are evolving strategies against that control agent, so drug resistance, for example. Depending on which type of disease, you might be interested in case finding. So it could be, let's say, a particular case of female genital schistosomiasis, or it could be a case of human trypanosomiasis. But also we kind of think of epidemiological design. So I think all of that is very familiar to us within our kind of daily jobs, but around it is probably where you bring in the One Health dimension. So for like schistosomes, the top one, climate change, water sanitation, urbanization, water disasters, cyclone flooding, people moving, those are really strong dimensions which crosstalk into the center. But also knowing if you go around the more human, specific or human creations, healthcare systems, access to primary care, global economy, shortage of praxiquantel, who produces it, for example, education, a variety of health aspects in that, but also knowing that a lot of schools are the platforms to deliver deworming drugs. And as many of you know, conflicts and human rights are one of those packages which do hamper some of the activities that you do in terms of public health, poverty. And then in the animal health dimensions, obviously food security, livestock production, that is probably where most of us are most familiar of diseases of livestock or wildlife are spilling over into human transmission or emerging. Anyway, that's the kind of graphic that we often use to describe the One Health space. So a few years ago, we used that graphic and we wrote an article with colleagues in Harvard and Georgetown University, mainly because we kind of alerted to the fact that with COVID, which changed a lot of things, it also changed the way in which we engaged with communities on the ground because of, social isolation and lockdowns and so forth. So it really did change the landscape. And we're still trying to best understand how to start up a lot of public health campaigns. But I suppose the thing that it taught us is that, diseases come from animals, basically, and they can spark off and cause a lot of global chaos. I'll just go back a second. So talk's really going to be on schistosomiasis, but I'll drill down to it. So the top of that slide, schistosomiasis has a bit of a PR disaster at times. It can be called bilharzia, which might mean that you're looking at a general type of infection, or it could be called snail fever, depending, it's an American terminology that's used. But UGS is a new terminology, urogenital schistosomiasis. It used to be called just urinary schistosomiasis, but UGS gets split up into gender-specific terms, so male genital schisto, MGS, and female genital schistosomiasis. Then, of course, we've got intestinal schistosomiasis. So that's the kind of like classifications we have within schisto on the broad. And it's interesting to know that this year, you perhaps didn't realize it, but it's the 200th anniversary of the birth of Theodore Bilhars, who was the discoverer of these schistosomes in Cairo, Egypt, around about 1851. And there's a lot of research since then, but it was in 1915-16 that they understood that it was transmitted in freshwater by freshwater snails, and that brought together the environmental health aspects together with the clinical medicine. So luckily, I'm often travels again going to Germany week after next to go to Tübingen, and there's going to be quite a large gathering over 3 days of people talking about various aspects of schistosomiasis. And I think it's also open, so you could probably join online if you so wished. So going back, most of you will probably be familiar that one of the species, Schistosoma hematobium, is responsible for urogenital schistosomiasis, and the other one, Schistosoma mansoni, responsible for intestinal schistosomiasis. Urogenital is associated with the urine, intestinal with the stool. And it's caused by large worms, adult worms, which live inside the blood vascular system. So it's unusual, called blood flukes. And they make the blood their home. So they can really, circumnavigate a lot of immunological responses to them. And indeed, they live in your blood because that's where they want to be. The dioecious, so the males and females, the female devotes itself to producing eggs. The eggs are very cytotoxic, and they're the problem with the schistosome is it needs to get out of the body, at least the eggs do. So the eggs are able to digest tissue and exit from either the bladder or either the bowel and burrow a hole and go through to either the urine or stool, which get released when people go for ablutions and other bodily functions, for example. The worms themselves are not monogamous. They change partners and they can form different cross-specific pairings. And then the image on the right is a blood vessel of a mouse experimentally infected with schistosomes. And you can see like a tumbled ball of maybe 5 or 6 worm pairs. The worms are very aggressive amongst themselves and they will steal mates from each other. And also they will change partners as well. So there's a lot of interesting breeding biology. It's only relevant for a later topic when we're thinking about the emergence of hybrid schistosomes, but it's just to point out that there's a very complicated sexual biology that goes on within every infected person. And that can go on for anything up to 15, 20 years, depending how long that infection's going without a praziquantel treatment. However, that's the simple story. That's what you find in the textbooks. This is what you find today, which is really why it's an intriguing challenge. This is a urine filter. And for those of you who know a little bit about the schistosome eggs, the eggs are typical of the species. And first of all, when you see this egg on the right-hand side, you can see the lateral spine, which makes it Schistosoma mansoni. Now, that's unusual. Why should a mansoni egg be coming out in urine? That should normally be coming out in the stool. This is the traditional hematobium with the terminal spine. That's in the right place. No surprise there. It looks morphologically typical. But this other egg on the top, probably about twice the size of the hematobium, still got the terminal spine, diamond shaped. That's a different schistosome. So this person has actually got at least three different species in them as a triple infection. And the mansoni egg is ectopically excreted in the urine. The Mattii egg, which we'll describe later on, is typically found in the urine as well, but we're also finding that it gets a lot of them in the stool, as does the hematobium. So this division of, what goes in the stool, what goes in the urine is either starting to break down, or the more we look at it with a variety of genetic markers, it seems to be a bit of an artificial concept. It's very helpful to understand clinical disease, but from the biology, it's not as simple as it looks like. So these adult worms can mate up in different ways, and sometimes they can produce viable offspring, which might be the emergence of a new type of schistosome. And these hybrid schistosomes are really causing a bit of a problem. So hematobium, which is the main cause of urogenital schistosomiasis, was found by us in 2006 in Senegal as a combination with Schistosoma bovis. And that was the first time using molecular methods that we described. We thought it could happen, but we proved it in natural transmission in Senegal. And that created a bit of a problem, because it showed that transmission of hematobium wasn't just tied to people, it could be tied to cattle. And also new types of schistosomes were appearing through hybrid form, which later probably explains why this type of hybrid exported itself and started natural transmission in Europe, as you'll see. So this bovis caused quite a stir because it was found in natural transmission on Corsica. So travelers that had never been to Africa caught urogenital schistosomiasis on Corsica, and it was described by French colleagues. We encountered the same bovis hematobium hybrid in Malawi, and we found it, funny enough, down in this place in the Sanji, which I'll talk to you a bit more. But also we found another variant in Mangochi. So these are photographs of eggs that you find from children's urine in this part of southern Malawi. And just noticing that typical hematobium eggs have got a size length type of distribution. These atypical ones are much larger, as like you saw in the other image. And from a morphological perspective, you'd say they're either very atypical hematobium eggs, or it is not hematobium at all. Interestingly, when we genotyped those eggs, we showed that it was actually, some of them were Schistosoma bovis hematobium, but also there was this Mattii hematobium hybrid as well. And this Mattii hematobium hybrid is intriguing because the colleagues, Tina Howes, who helped us describe the hybrids in 2006 in Senegal, being Belgian, was collaborating with the Institute of Tropical Medicine there in Antwerp. And they were made aware of travelers to South Africa talking to John and Bhavani about this. But this got published in the clinical infectious diseases. There was a group of 34 Belgian tourists that were in having water contact in the Mapelangela area. And when they went back to the Belgium, they had acute schistosomiasis, and those infections were worked up, and it turned out they seemed to have caught either a mixed infection, which is a hematobium and Mattii, or they had a Mattii hematobium hybrid bona fide. So this prompted me and some of my other colleagues to ask this question, pose this question, which for the younger audience, when you have a question saying, will all hematobium hybrids please stand up? Some people might think of M&M and Real Slim Shady, but this was a homage to a colleague of mine, Wendy Gibson, who posed the same question, probably about 1986, about will the real Trypanosoma brucei gambiense please stand up? And it's just to kind of reiterate the fact that the taxonomy of a lot of these worms or parasites is a bit more dynamic than we think. And for this particular hematobium, there must be a lot of lineages which are present, which are not hematobium proper, but introgress genetic forms. The reason why that's important is because when we think of long-term control, hematobium would have its own cycle, which is like unique to people. And then Mattii, which is well known in the veterinary circles, would be, say, specific to cattle. But with these introgress forms, you have this new setting where the schistosome or the lineages that it produces can either go into human hosts or either livestock hosts, but at the same time, this generates a tremendous amount of biological diversity and also control refugia. So in other words, if you have a campaign which is only treating people and you're omitting treatment in animals, for example, you know that you're not going to have transmission control. There's always going to be a gap in there. But this biodiversity creation is really important because schistosomes are unusual. They can form very viable introgress forms, and the hybrids can actually have greater egg production, and they can also utilize different snail hosts. So it's a really unusual driver which is specific to the biology of these worms. Another thing to bear in mind, in Malawi, there's a change in the snail fauna, which has also sparked an epidemic or an outbreak of intestinal schistosomiasis in an area which was once free from the disease. And we'll come on to that unusual scenario in Lake Malawi in a second. This is one explanation of why we have these triple infections quite often. So this brings on to the second half of the talk, which is really looking at the epidemiology of schistosomiasis. So this is a view from Southern Lake Shore, children in the water swimming, people fishing, hotels, recreation, so forth. Beautiful place. And then the next is urine samples that we get from the cohort that would be following. I should tell you a bit more about that later on. Some of them, for the very keen eyes, might be matched up with a fecal sample, but we saved a lot of time at the start of our surveys just doing urine sampling because we thought we'd find the majority of the schistosomes in the urine, as you saw from that Mantsoni, Mattii, and hematobium urine filter. The interesting thing here was we had a funding from the Wellcome Trust, which gave us four and a bit years, and it was a joint investigator award with my colleague Jane Lisa Messiah at Blantyre. So we had two study sites, which goes back to where we first found these unusual hybrids. So we have Mangochi district and the Sanji district. The background map is just a general control map of intestinal schistosomiasis, which is a bit more patchy than urogenital, but it's just to highlight that the lake wasn't really thought of having natural transmission, but we'll come on to that in a second. Anyway, this is a project. It was a multidisciplinary study. We had people recruited from two villages, about 1,200 in each village. We expected them at 3 time points: baseline, MDA, first follow-up, MDA twice, second follow-up, as you see in this little Gantt chart here. So we had a human work stream, had a snail environmental sampling. We looked at 7 locations in Mangochi over 2.5 years, looking at the snails, mainly because we wanted to genotype the infections before we screen the people, but also knowing that when people get treated, they get reinfected. So could we actually find these unusual schistosomes in the snails before people got reinfected and so forth? And then at the same time, I'll come on to this as a major part of the talk about the infections in cattle. So for those of you who have to get funding from large-scale grants, over the years, this is a bit of a cookbook. You have a pilot stage, you have your experimental, your surveillance phase, and you have a dissemination phase, or like result sharing. So we had a a UK dissemination meeting in February, which was the Royal Society, and that gave a lot of attention to the disease. So from the highest academic society in the UK, people were starting to get interested in this problem. Anyway, why Malawi? Well, this is the reason, is because it's a big tourist attraction. A lot of people go to Malawi. There's a big disease risk in Malawi. I would say probably about half the population of Malawi are at risk of some form of schistosomiasis, and probably about half of those have probably got active schistosomiasis, so they've got egg excretion in either stool or urine. Hematoma is pretty dominant, but also I wanted to point out that it flags up on the UK radar as well with travel related. And one of the reasons why people go to Lake Malawi is just basically for looking at fish. They do a lot of scuba diving, for example, and also it's convenient for sailing. And it's a bit of like an inland sea for like tourism. So it generates a tremendous amount of income from Malawi. So going back to that travel related thing, colleagues at the HTD down in London published a paper a few years ago, and it's just to point out that top of the pops of the people that they saw in clinic were from Malawi, and noticing that most of them had hematoma eggs in urine, and a smaller proportion had stool probably intestinal schistosomiasis, schistosoma mantsoni in their stool. So this is published by Cordelia. And I just point out that you probably recognize some of the names, Peter Chiodini, but also this chap, Chris Whitty, who I don't know if you realize he became the chief medical advisor in the UK just before COVID hit. And unfortunately, he was lumbered with perhaps one of the biggest public health challenges in our lifetime. But he was a very erudite infectious disease clinician with a lot of tropical disease experience as well. Chris is still the chief medical officer in the UK. I think he steps down in about a year or two. But as you know, COVID changed a lot of the way in which we had public health in the UK. Now, I'll just step back a second. Why Malawi? Well, interestingly, so schistosomiasis wasn't always endemic in the lake, and it was only until about 1985 that, well, maybe it was endemic in the lake, but there wasn't much surveillance for it. But it started to come up on tourists. So tourists to the UK coming back, American tourists and so forth, they started to notice that they had urogenital schistosomiasis, probably from scuba diving up in Cape Maclear. There was also the connection that around the lake, you've got these small water habitats which can have a different snail called Blindness globosus, which was not in the lake, it was on the periphery, but that sparked a large-scale study on Cape Maclear by Danish colleagues from the Danish Bilharziasis laboratory, and then they showed that not only do you have these snails around the perimeter of the lake, but you also have a deep water snail called Blindness nyasanus, which is found on exposed sandy shorelines down to the depth of maybe 15, 20 meters. So the old advice of swimming on beaches, which are largely artificial to start off with, was kind of incorrect. So this snail, for some reason, started to transmit schistosomes where it once didn't, or perhaps it once wasn't noticed. It's unusual, this mollusk, because it's the deepest one in fresh water, can go down to 100 meters. So it's a very unusual snail. Anyway, I suppose when we now, in hindsight, we probably think one of the reasons why the schistosomes started to utilize this snail is probably because of some type of hybridization going on, and then intragress forms expanded their intermediate host range and then started to exploit another snail, which was common in the lake, but in a different microhabitat. So I got really involved in Lake Malawi in about 2016, supervising a PhD student, Seki Kayuni, clinical doctor. He was interested in male genital schistosomiasis. So he did a study cohort of looking at male genital schistosomiasis. But at the same time, we were doing snail surveillance, looking for Bolinus. But surprisingly, we also found Biflaria, which really sparked up another interest. And as I said before, there's an outbreak of intestinal schistosomiasis unfolding in Lake Malawi, in addition to the chronic urogenital schistosomiasis. So those were the justifications that enabled us to get funding from the Wellcome Trust. It was called HUGS, hybridization in urogenital schistosomiasis. And I'll just draw your attention to two studies within that, the animal stuff, so looking at the cattle, and then a little bit about male and female genital schistosomiasis. So the cattle work, we've got a website, LSTM/HUGS, and you'll see what we're doing. We put up a lot of blogs pretty much once a month, showing, for example, Alex talking to the British Association of Veterinary Parasitologists. bit about work that we've done on FGS, MGS, but this is an important one. There's a strong connection between MGS and FGS with HIV transmission, and one of our colleagues, Augustine Choco, had done a randomized clinical trial of access to oral HIV testing as well as access to praziquantel treatment in fishermen. And that's an unfolding story because there's a crosstalk, as you see later on, of FGS and MGS with HIV transmission, as I'll elaborate. And then latterly, in 2004, in November, we published a paper on goat-related transmission of Schistosoma mattii. Anyway, if you have a look at it, it gives you the idea of like the communications that happens on a large grant. And then I suppose most recently, we've been doing some snail mapping, predictive mapping. This is me putting GPS bracelets onto cattle, which I'll tell you the story about in a second, and then that AMR in snails. And also, because we were doing a lot of fecal sampling later on, we were screening for other parasites, and this flagged up Strongyloides fullerbornii in children, as well as in their vervet monkeys. We might pick that up later on if anyone has questions about Strongyloides. So this is what it was, hematobium hybrids, please stand up. And I suppose the question mark here, you're in a complicated transmission landscape picture. You've got people, you've got livestock, cattle, you've got goats, you've got sheep. You've probably got other things, other animals, even baboons that go around the freshwater habitat. What is the transmission? What parasite do you find in which host? So the bovine schistosomiasis was an easy one, simply because there's absolutely no surveillance for bovine schistosomiasis and malaria. So the veterinary sector is completely blind to that disease. This was a paper that was the fruition of a bit of work, and I'll talk you through its major results, but it was published in One Health. But it brought together the bovine surveillance with the genotyping of the worms to reveal what type of parasites were where, and also, as I'll talk to you about what type of options we might have to control bovine schistosomiasis as an input, zoonotic input, into urogenital schistosomiasis and Lake Malawi. So the next, for those who, this is like kind of classic One Health, or at least veterinary parasitology. I suppose food production animals are particularly unusual because if you get access to a slaughterhouse, you're able to dissect the carcass and find the worms. So this is myself and Alex inspecting entrails of a cow. And then if you look through the mesenteric veins, you're able to pick out the worms. You can genotype the worms. So this is great to actually see the adult worms. You can tease them apart, males and females, work out, is it, this type of species with that type of species. And also you can hatch the liver, you can hatch the stool from that animal. But the problem is you don't know where that animal has come from. So where the animal is slaughtered is not the same as location where it was raised. And a lot of slaughterhouses are like central locations where animals are exported in. So the geographic signature gets a little bit confused, but it's still quite a good thing to do. The flip side, and I'm not sure if this is going to play, but this is where you do a fecal hatch test. So you take a fecal sample from a cow, you simply kind of mix it, filter it, put it into water, and then observe it under the microscope. And if you're lucky, I think it will play. Actually, I'll tell you what's in the sign off. We've been doing this today quite a bit, and then that's what actually one of the colleagues in the lab was saying it looks like Casper the Ghost, but that is what they look like under the microscope. You can see them swimming, and if you use bottled water, it's a good technique, and because it's, where the cows come from, and also you can genotype. I'll just play that again. You can catch that Miracidia and genotype it with PCR, and then you know exactly what type of species it is, and you can also sex it as well because I'm male or female. Anyway, that's a really useful technique. It's a bit slow. It's like belt and braces approach, but combine the two, you get quite a lot of information. So this is what we did. Fecal sampling up in Mangochi, 120 cattle. Fecal patent prevalence was 67%, split across 3 different herds. If you go to the south, there's District Chikwawa, quite close to Blantyre, but Nisanji, where we were, interestingly, the distribution of bovine schistosomiasis there is very, very patchy. Long and the short of it is, if you compare north and south, there's a big division. So there's quite a big change in the landscape. There's a lot more bovine schistosomiasis on the lakeshore rather than down here. I'll just add the Shiri River flows southwards from Lake Malawi, goes around Blantyre, then comes down here, then goes out into Mozambique. Similar type of snails, not exactly the same, but it's a connected waterway. And then carcass sampling, bit of a lower prevalence, but we could genotype the worms, which is quite useful. This was one of the things. So we established that there was a problem in cattle up on the lakeshore, but we wanted to try and find what can you do about it. So where we were staying, right next door to where we were staying, there was a small herder which had 8 animals, which every day get grouped with another 9 or 10 and another 9 or 10. It forms a herd of about 30 animals, and then they walk around the location. So because this was very convenient, these 8 animals, we decided to do a bit of a proof of concept study, which was putting a GPS data logger onto them and then treating the animals and then seeing what their reinfections were. So GPS got put onto the animals, we were mapping them, and then 2 weeks later, treated the animals, 40 milligram per kilogram praziquantel, bottle fed. And then a week later, 4 weeks, 6 weeks, 8 weeks, and then monthly, looking at when the reinfections come back. I'd also point out that one of the smaller animals, I think it was that one, had a Hematobium mattii hybrid. The rest seemed to have mattii by itself, but the younger animal seemed to carry the hybrid, which is interesting. So those are the bracelets, put them around the cow. That's the movement that they have. And then this was quite a challenge. One of the reasons why we had to have very good community participation. We knew the owners, we had to change the batteries every week, and that was a real hassle to kind of immobilize the cow, change the batteries, but we got a lot of information. This is a graph of the number of miracidia that you can get in a hatch test. These are the animals. Just noticing that the younger animals probably have a slightly more patent infection than the older animals. Could be argued that there's some type of like partial immunity there, or simply that the animals are just bigger. So that's a bit of a debate, which is raising a question. Well, when we think of future control and treatment, it might only need to be in the younger animals because they might only have the hybrid forms. But we've got to kind of discuss that a bit more later on. Anyway, we did that. We showed that all the animals in that herd were infected. Treat them. It clears the infection very, very satisfactorily. 6 weeks later, the younger animals came up reinfected, 8 weeks a bit more, and then by 3 months, all animals were reinfected, but the levels of excretion were lower. So interestingly, it showed from the water contact, the animals have about 45 minutes exposure to the lake along the zoning. And as good fortune would have it, one of these animals actually gave birth to a cow 2 days after we gave the praziquantel treatment. So we monitored that animal as well, and that animal became patently infected 8 weeks later. So it's showing that there's active transmission and reinfections on that lake shore, certainly in the cattle. So we genotyped them, and as I mentioned, majority of infections were mattii. So those cattle are contaminating the shoreline with mattii, but they're also able to contaminate, at least the younger ones, with a mattii hematobium. And then another thing, if you looked at that paper, we did find hematobium itself in cattle in a different herds, quite minor, but 1%. But to say that hematobium cannot be found in a cow, well, that's not true. This is what the cattle do. This is where they sleep in the corral. They get out in the morning, they go out and graze. If there's grazing, then they go to be watered. And we follow some of the animals, and this is like some of them actually go into fairly deep waters, as you'll see in a second, which obscure the GPS signals at times. So they're crossing in water, and then their movements are really really contingent to this guy. He's moving around and grazing. And then I suppose my punchline is the life of a cow is very much like an academic these days. We just eat, we sleep, we walk, and then we repeat. But yeah, maybe it's one of those things which we learn a lot from studying other animals. So now I'm going to turn attention just the last 5 minutes on the zoonotic aspects of male and female genital schistosomiasis. And I'll just point out that we'd studied the human cohort at 3 time points, and then we actually, in the second and first follow-ups, we tagged a group of people that we wanted to do some clinical investigations of genital tract disease in quite greater detail. It's not something you can do very frivolously. It takes a lot of setup, but we'd implicated those people the year before, and we got set up. We were working with our consultant gynecologist and midwives, Dingazi Kumbweni. So this is just to show that the general prevalence, to make you aware that in Mangochi, Samama village, and the Mithawiri village in Isanji, the overall prevalence was about 50% egg patent urine. And after like MDA once, MDA twice, you can push it down, but it doesn't go away. And also, I just want to point out, if you look at macrohematuria and also microhematuria, even though you've been given out treatment, they seem to plateau around about 5 or 6%. So it's a bit unfortunate that the treatment that you give out is pretty much eroded by the reinfections, which is kind of confirmed what we knew about in the cattle. Anyway, that's a paper that could look at the other thing. I want to draw your attention to this. This is the clinical case that we had a particular woman, which we made a story about down in the Sanji. We've published another paper later on that we'll be able to talk about, but this is an intriguing case which might pique people's interest. So that individual was resident down in the Sanji. She'd often cross the river, go to work in the fields. She had a variety of gynecological symptoms, and I'll show some photographs, but we worked her up. We did a variety of testing, and it's just to point out that diagnosing female genital schistosomiasis is not as simple as it sounds. So urine CCA, urine filter and microscopy schistosoma over negative. So you could get misled straight away by absence of eggs in urine. Next one, if you do cervical vaginal lavage, swab, screen it for PCR, still negative. However, if you do a cross biopsy of a biopsy of the cervix where you see some pathology, you can see schistosome eggs, and you can PCR it as well. So anyway, it's a sign of a, what you might say, dry infection, but a morbidity signature. And I just want to turn your attention, you can find other STIs in there, but the HPV virus is associated with viruses negative. The reason why I want to show you that is because when you look at the clinical, presentation externally, this woman had evidence of what you might say external warts, but they're not warts. They're probably old external granulomatous tissue caused by bilharzia. So you can see some objects down here and up the top right. But if you take a biopsy from the cervix, you can see it's festooned with like eggs, mainly calcified eggs. So this woman is a very clear case of probably a chronic FGS, which is fairly incipient in being able to detect it with kind of like, standard parasitological methods, unless you go to crush biopsy. Now, this is the thing I'm really happy about because I was able to show that microscopy is still very useful. A lot of colleagues that use CVL will spin it, sediment it, and then use genetics to try and detect, various parasites and pathogens. And we thought, well, actually, just do a simple thing, resuspend the sediment and look for eggs. And you can find schistosome eggs. You can find viable schistosome eggs in the CVL sediment. And also, you can get a lot of them. So this is a graph just saying for some women, they can be excreting up to like 50 eggs in a single CVL sediment, and some of them might be less, but that's like egg excretion across, the vaginal or the cervical surfaces, and allowing the eggs to get out from the body without going through either the stool or the urine. And this is a thing which I think is quite important, and not many people have connected it up. There's a lot of like public health, like menstrual hygiene management, access to sanitary products. But as we saw before, a lot of those sanitary products don't get, disposed of correctly. And also, if people wash some of these things, there's a transmission route there for parasites to get in and out. And I just want to point out that there is a connection here with menstrual hygiene management in people that have got, female genital schistosomiasis. So something to think about. So this is the surprise. I just want to finish off. We spent a lot of time using urine because we wanted to, scale up our testing because we're looking at, 2,000 odd people, and it's not easy to get a lot of fecal samples from that number of people. But in the follow-up, we decided to do a little pilot study with a PhD student who was looking at fecal samples mainly for AMR markers. And we were surprised at a follow-up one. In the fecal, we see, interestingly, that's the hematobium egg with a nice spine at the top. This is a lovely little Mansonet egg with the lateral spine going that way. Those are the typical ones. But look at the other two. This could be an atypical, don't know, hybrid, or could be like a bovis-like egg. But then you've got the typical Mattii-like morphology. So you can see morphologically, there's quite a lot of diversity. And then in the second follow-up, we had two patients that we hatched Miracidia from fecal and then hatched Miracidia from urine and compared what we got. And we were really surprised. So two patients, one was a 18-year-old boy, one was a 11-year-old girl, forget which one was X and Y, but that's what you see in the urine. So you'll see hematobium dominant, but you'll also see a hematobium mattii Miracidia in the urine. Same for the other patient, patient Y. But if you look at fecal sample from those two patients, same things are present, but the numbers of the hybrid of Mattii hematobium hybrid is massive. And also there's Mattii proper. So this person, what you see in the fecal is more Mattii in stool than Mattii in urine. So there's something going on there. But the one patient-wise is really unusual because of this. We also found a Mattii-Mansoni hybrid. Now it's probably a dead end, but the fact that we're finding it, it has been observed in the laboratory, but it's never been found in nature. But just look, this person's got Mattii stool, Mattii-Mansoni hybrid, Mansoni eggs, Hematobium mattii hybrid, and then Hematobium eggs and stool. So intestinal schistosomiasis there is caused by a pretty complicated mashup of parasites. And I don't know what the pathology is going to be in terms of hepatosplenic disease associated with that. It's an open question. So we kind of wrote 2 little summary articles, which you were very welcome to look at. One was just drawing attention to Mattii and its zoonotic potential, and then the other one was the male genital schisto, which I haven't really talked about much greater detail other than giving you that FGS case. And then I think maybe it's the way to celebrate. It's a world first in a negative sense. Do you know of any parasites or pathogens that are zoonotic which cause genital tract disease in people? I think that's a bit of a mind shift, which is difficult for the SRH community and even parasitologists in general to think that, you know, gynecological disease or male genital tract disease can be caused by a parasite that comes out of a cow. I think that's quite a food for thought. So, summary, schistosomiasis, at least what we know of it in Lake Malawi, is quite unusual, some surprises. You find a lot of triple infections, perhaps quadruple infections if you include the hybrids. And if you give out mass drug administration, it has an impact, but not as much as we would like. But also the Mattii, the prevalence of Mattii stays pretty static across all time points, which is probably because zoonotic transmission is kind of constant there throughout the year. So you're always going to get zoonotic inputs. So this is one of the reasons why we need to think about what type of control is possible, you know, fencing or access to treatment in cattle, even cattle vaccines might be up there again. So looking at snail dynamics, how about we think about livestock control? And this is important because when we think of like elimination of schistosomiasis, especially in areas where there's good, you know, control, you know, zoonotic sources, like for example, on Corsica, that was a surprise for the schistosomiasis community. So we should be alert to the fact that schistosomes can change. Just to finalize, final slide. One of the reasons it's a great pleasure to be here in South Africa, working with John, Charlotte, and Bhavani. And Bhavani's hopefully going to be doing a PhD, looking at better surveillance for Mattii, which builds on a lot of, you know, historical stuff, but that historical stuff needs to be reevaluated again. So hopefully looks at urine sampling, might start to think about other aspects of embellishing that. But we had a lovely little pilot, you know, to see what type of snails and parasites we can find. So wish Bhavani the best over the next years. And I think she'll be incredibly well placed to make a really important contribution to understanding schistosomiasis in Southern Africa. So that's me. And then I hand over to you for questions. And once again, thanks to John. John was marvelous because this presentation wouldn't be half as good as it would have been. He lent me his computer over the weekend, and because I was computerless, so I was able to try to peak this to the general interest. So thank you very much.
Wow, fantastic stuff. Really, really interesting, and I'm sure most of us have learned a huge amount from your talk. I think the hybrids are fascinating. So while people are typing their questions in the chat and things like that, I just wanted to kick it off with saying, I mean, do we think this is a fairly new phenomenon, or is it just that we've only discovered it because we're now using more molecular methods? I mean,
bit of both, Jeremy. Bit of both, because historically, you could keep some of these isolates in the laboratory, and you could make these artificial challenges. I think for certain combinations, I mean, there are about 6 different species of human schistosome. So this Hematobium mansoni by itself is not strictly true because you've got intercalatum, you've got guineensis, you've got this Mattii as well. And depending on what the combinations are, some of those like hybrids are perhaps more likely than others. But you're right, the molecular techniques are helping us to detect it. But at the same time, I think there's a lot more close contact of agriculture with, you know, human populations, be it from water impoundment schemes or from like, you know, cattle herding, which is largely unfenced in a lot of areas of Africa, which brings people to, very close contact. I think we have to be very strict with this. You should try to restrict cattle water contact with environmental sources that are going to be used by people. That's going to be incredibly difficult to achieve, but over the years, I think that's increased as people have used livestock as like food security as well as, financial resource for a family. So I think it's a bit of both. I think certainly that Bovis hematobium hybrid that was never been found in Corsica, so that is a real surprise. And I think, and when we found it going south in Malawi, there's a lot of plasticity in these hybrids, and we don't really understand where they might spark an epidemic or, an outbreak.
Yeah. I'm just looking through the chat. I mean, there's a couple of questions on the mechanism of hybridization. So is it presumably when one, say, snail host gets infected with both species simultaneously, and then does that hybrid then have to sort of, I mean, how does it propagate beyond that? Because obviously there's only one hybrid now. So does it
Well,
go back
actually,
to the species?
it's interesting because I suppose if you remember, most flatworms are hermaphrodite, but schistosomes are unusual. They have a male and a female. So the hybridization typically plays in the definitive host where you'll have, let's say, a male Mansoni and a female hematobium, and they can form like a cross-specific worm pair. They will fertilize the eggs. There's also parthenogenesis here as well, which complicates the factor. But if you autopsy an animal, you can tease the worms out and you can find these mixed worm pairings. That's basically where they fertilize. Now, these eggs that come out might be introgressed or hybrid forms. If they haven't got the right machinery to infect the snail, they die out. The snail's unusual because you have like asexual reproduction. So the horror story might be you get a new hybrid which has got a new intermediate host spectrum. One mirror city getting out can expand itself in the environment because it's compatible with the snail. And it's just a matter of chance if whatever definitive hosts that come back to use that water are able to get a sufficient dose to establish that infection. And then you'll get like a transmission effect going on, through time. It's well known in the laboratory. You can do the experiments. You can't do all combinations. So this is one of the reasons why I got very excited by that Mansoni-Mattii hybrid. I think that hybrid is incompatible with either Bolinus or Bienfalaria because it's just mixed up its genes too much. But the other ones, which I mentioned, Hematobium and Mattii, they can probably host switch within Bolinus, and that's really important because the distribution of the snails is quite heterogeneous. And if you've got a parasite which has got a new type of intermediate host spectrum, they can colonize environment that they could never have colonized before. But it's in the definitive host. And also, as a parasitological joke, how old can a schistosome be? It's like it can be as old as its host. So, a schistosome worm can have like 30 years inside someone, swapping mates, parasites, blah, 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 with females and whatever. And then, there's this enduring phenomenon of being able to form these hybrids in the definitive host for a long time. And that's a real trick that parasite has got. And yeah, I think that's a clever thing it can do. Time for a bit of cake. Next question.
And then I guess so sort of related to that, because obviously it does seem to offer advantages in terms of host range. And I think you said that some of the hybrids produce even more eggs. So what's the downside? I mean, why hasn't evolution just made them all effectively hybrids by now, if you know what I mean? In other words, why?
Well, I've got a good answer to that, Jeremy. My lifespan is only 30, 40 years more than what I've got at the moment. You might find that evolutionary scenario is going to play out over, decades, 100 years. So if, 200 years time, there might be no such thing as Schistosomiasis, hematobium. It might be a new mashup or something else. But, history of humans, human population expansion, all that stuff, that's a new dynamic, maybe only 100, 200, 300 years old. And I think evolution can't necessarily speed itself up to get around that. But 200 years time, that question might be very different.
Fascinating. And do we know anything about any clinical differences between these? I mean, do they present any differently, or is the treatment any different, or do we think so far it's hard to tell?
Hard to tell. I think I would like to see more studies on the secreted compounds that you get out of an egg. The egg antigens or toxins you get in Mansoni are very different to hematobium. hasn't been enough research done on like Mattii, but I can envisage a scenario if you get a lot of, you know, hybrid eggs, you could get a slightly different immune reaction to it. Whether or not that's highly immunopathogenic, don't know. I think it also depends on the number of worms you're carrying. So if you have a heavy infection, it might be augmented a little bit by the hybrid, but you know, the bladder cancer, the fibrosis of the bladder, even this thing about trying to find, you know, schistosome eggs in hematobium eggs in fecal, you might find that there's some other, you know, liver disease which we haven't fully got a handle on either. But I suppose hybrids are probably more important, not from the individual, you know, clinical effect. It's more of like the community control and what that problem poses for transmission control.
And then I guess one of the last questions, but one of the people in the chat's asking about just treatment of the water. Could something be done in terms of reducing the burden of disease that way?
Yeah, this is
Lake
a little bit of
Malawi,
a joke. This is a bit of a joke. We pollute our waters with a hell of a lot of plastic and rubbish, and yet we can't use snell control methods. So there are chemical molluscicides that you can do, and people in environmental health will do that in small streams. In Lake Malawi, it's just it's impossible. It's too big. Also, it's a fishery. So you would blow probably the whole budget of chemotherapy on snell control over like maybe a 10-kilometer area. So it's just not cost effective. However, if you want to stop the spread of a snell into irrigation scheme or something like that, snell control is incredibly important, like chemical control. But I'm afraid we have to be better stewards of our environmental, you know, water environment to kind of like stop like schistosomes from gaining that upper hand of being able to exploit environments which we have largely created and which allow other people to be affected or livestock. So it's a little bit, we kind of like create the problem, we try to solve it. But yeah, it's a little bit beyond us with current tools, I'm afraid.
Bhavani, I see you've got your hand up. Do you want to ask a question?
Thanks, Jeremy. It's Charlotte sitting here with Bhavani. Just to say very briefly, a massive thanks to Russell for doing this talk today. It was absolutely brilliant, and we really look forward to the future, to collaborating on all things schisto. And just for the audience to know that at the NICD, we are doing a laboratory-based surveillance for schistosomiasis. Nested within that is what Russell mentioned about Bhavani's PhD. We're looking at molecular epidemiology. But I think we're just starting to scratch the surface, and there's lots of work for us to do to try and impact on public health in terms of schistosomiasis. So anyone from the laboratory or clinicians, if you have any interesting cases or any unusual looking schistosomes in your labs, please reach out to us. We've got reference testing. We are setting up testing for hybrids, etc. And we'll share our interesting field study and other surveillance results as we go along. Thanks very much again, Russell. Thanks, Jeremy.
No, it's been a real pleasure. Thank you very much for the hospitality. It's great to meet a lot of colleagues and also, you know, as they're going to the Kruger Park, me and a lot of other parasitologists, it's really important to maintain that interest because, you know, schistosomiasis is really, really common and yet doesn't get given the priority it maybe should have.
Wonderful. Thank you so much. Really appreciate it. And Professor Bloomberg says we all want to come on your next field trip. So
Oh, thank you, Lucille. And then also, yeah, come to Liverpool soon. I'll enjoy to see you there. Brilliant.